Good morning, students. This is your teacher Neelam Elawat, and we are continuing with the same chapter, that is molecular basis of inheritance, and this is our lecture number 6. Now, yesterday we talked about the replication process, isn't it? Now, today I'll be dealing with how did we came to know that the replication of the DNA is a semi-conservative. Okay, so I will talk about the Meselson and Staw experiment today. So I hope you are ready, students. So let's start, everyone. Now we see the replication is a process by which DNA produces the same copies of itself, right? And this occurs in the S phase of the cell cycle. We have talked it about yesterday, so a little bit of revision. And the Watson and Crick, they propose the double helical structure of DNA. Okay. The structure of the DNA was proposed by the Watson and Helical, and they said that the DNA is a double helical structure. And in the same way, they had written their a statement. It says, it was not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Clear? This means they suggested that the DNA replication is a semi-conservative method. DNA replicates itself through semi-conservative method. They said that the two strands of the DNA, they will separate. One will act like a template, and there will be production or the synthesis of a new DNA strand which will have the bases complementary to the template strand. And the new DNA molecule which has been formed will have one strand of the parent, and one strand is formed new. Okay. But apart from this, there were three possible mechanisms for the DNA replication. There were three basic patterns. Okay, let's discuss those three basic patterns. Suppose kariye this is the DNA. The first model that we talk about is called conservative model. ठीक है, 3 मॉडल्स हैं. फर्स्ट जो हम बात कर रहे हैं वो कंजर्वेटिव मॉडल. द कंजर्वेटिव मॉडल सेड द डीएनए मॉलिक्यूल जो हमारे पास है, पेरेंट सेल में जो डीएनए मॉलिक्यूल है, ठीक है, इट विल नॉट सेपरेट. ओके, दिस डीएनए मॉलिक्यूल विल एक्ट एज अ टेम्पलेट एंड अ कंप्लीटली न्यू डीएनए विल बी फॉर्म्ड. क्लियर? एक नया डीएनए बनेगा कंप्लीटली, जो हमारा ओल्ड और यू कैन से पेरेंटल डीएनए है वो सेपरेट नहीं करेगा, but this will only act as a template and a new DNA will be made. This model is called a conservative model. ठीक है, जिसमें parental DNA separate नहीं होगा और new DNA, completely new DNA will be formed. Both strands of the DNA will be new. The second model is called the semi-conservative model. The semi-conservative model says that the parental DNA will separate. Okay. The parental DNA will separate and both strands will act as the template and new DNA will be formed. So the new DNA that is formed will have one parental strand and one newly formed strand. तो आप देख लीजिए दोनों हैं इसमें, एक strand नया है, एक strand पुराना है. Same here also. ठीक है, this model is called semi-conservative model. Then there was a third model which was called dispersive model. What it called? Dispersive model. क्या कहते हैं इस मॉडल को? Dispersive model. Now this dispersive model says that new DNA will be made. जो नया जब फर्स्ट रेप्लिकेशन होगा इस DNA का, from this DNA, this parent DNA, when new DNA will be made, there will be a mixture of old and new DNA in both of them. देखिए, some portion is of the old and some portion is of the new. क्लियर? जो नया डीएनए बनेगा उसके अंदर कुछ पोर्शन पुराने डीएनए का होगा, कुछ पोर्शन नया बनाया जाएगा. सो दिस मॉडल वाज कॉल्ड द डिस्पर्सिव मॉडल. बट लेटर ऑन द Meselson Staw proved it experimentally that the semi-conservative model is being followed during replication. So Meselson Staw जो हमारे साइंटिस्ट हैं, they proved that out of these three, the DNA only replicates or uses the second method, that is semi-conservative model. Clear, everyone? So your conservative model, the original DNA will remain intact and entirely new molecule of DNA will be formed. Dispersive model, the two DNA molecules will have some part of the old DNA and some part of the new DNA. And the semi-conservative replication says that the two strands of the parent DNA will separate, one will act as a template, and you can say one old strand, one new strand will be formed. Now let's see the measles in straw experiment. We'll talk about this experiment, isn't it? So let's see this. Measles in straw, they proved it experimentally. What they did, they performed the experiment on E. coli. Okay. Now first of all, they grew the E. coli on a culture medium that was having ammonium chloride 15. The nitrogen was nitrogen 15. Okay, the medium was having nitrogen 15. Now this nitrogen 15 is a heavy isotope. तो ये heavy होता है. So when it is being incorporated in the DNA, DNA will also become heavy. Okay? Clear है? Then both the strands of DNA will have N15, N15, N15 at the 0 minute. 0 minute means initially हमने sample के अंदर N15 डाला है. So DNA जो है DNA के अंदर N15 मौजूद होगा. ठीक है? Then this DNA will be extracted, removed and will be subjected to cesium chloride density gradient centrifugation. After some time, after regular intervals, they will be subjected to cesium chloride centrifugation method, and depending upon the density of the DNA, they will separate at different levels. अगर heavy है तो नीचे मिलेगा. If it is intermediate, in the center. If it is lightest, it will be on the top of the solution, isn't it? Now after this, once they have been grown in N15, the bacteria, they were shifted to a medium which has N14. Now this N14 is a lighter isotope of nitrogen. So wherever in DNA N14 is present, the DNA will become lighter. Wherever N15 is present, the DNA will become heavier. And wherever both are present, the density of the DNA will be medium. Clear? So after DNA extraction, again there will be cesium chloride centrifugation after 20 minutes. Okay? After 20 minutes and then after the 40 minutes. And we'll come to this experiment now. Suppose सबसे पहले हमने क्या किया कि N15 के मीडियम में हमने E. coli को ग्रो किया. ठीक है? When we have grown the E. coli in N15 medium, तो हम 0 टाइम पर हम उसका सैम्पल टेस्ट कर रहे हैं. ठीक है? So when we are testing and subjecting it to cesium chloride centrifugation method, so we observed that the DNA is present at the bottom of the test tube. ठीक है? Bottom में present है क्योंकि जो N15 है, it is a heavier isotope of nitrogen. तो हमारा DNA भी heavy हो गया है. ठीक है? Is it clear? So we are indicating this DNA with blue, completely blue. Blue DNA means it is having the N15. Okay, it is having the N15. We can match with the solution of the color. Color of the solution you can also match. Here it is blue. So the DNA is completely blue. Okay. Then these bacteria, they were shifted to solution which has N14. ठीक है? तो सबसे पहले हमने क्या किया, बैक्टीरियाज को N15 में ग्रो किया, फिर उन्हें 0 R पे सेपरेट किया. तो DNA will be obtained at the bottom because it has the heavy, because the DNA is heavy. Okay, so the DNA is present at the bottom of the test tube, and the color you can see it is of blue color. Now second, we are transferring it to the medium which has N14. So N14 को हम red color से indicate कर रहे हैं. ठीक है? And we left it, the bacteria were left for 20 minutes. So we have taken the sample after 20 minutes. अब हमने 20 मिनट के बाद इस sample में से we have taken the sample from the solution. And again we have subjected to cesium chloride centrifugation. जब हमने centrifuge किया, ठीक है, जब हमने centrifuge किया है, now it is present at the medium level. देखिए अब हमारा जो है medium level पर मौजूद है. मीडियम लेवल है, डेंसिटी का जो है मीडियम लेवल है. मीडियम लेवल का मतलब है नाउ इट इज हैविंग N15 ऑल्सो एंड N14 ऑल्सो. And if you remember, yesterday we talked about that E. coli takes 18 minutes for one replication. याद है आप लोगों को? हमने कहा था E. coli जो है 18 मिनट लेता है अपने आप को रेप्लिकेट करने का. So we are taking the sample after 20 minutes. Means one replication cycle has been completed. तो अब अगर हम कह रहे हैं कि डेंसिटी मीडियम है, डेंसिटी मीडियम मीन्स कब हो सकती है डेंसिटी मीडियम? When one strand is old and another is new. So in the DNA, one strand is old, another is new. Okay, this proves that the replication of DNA is a semi-conservative method. ठीक है? Then after 40 minutes, we again will take the sample. Now after 40 minutes means now 2 replication processes have occurred. जो E. coli है वो 2 बार replicate हो चुका है अब. ठीक है? अब हम देखते हैं तो हमें दोनों सैंपल्स मिलते हैं, हैवी भी और लाइट भी. हैवी ऑल्सो एंड लाइट ऑल्सो. क्लियर? नाउ लेट्स सी द DNA. अब देखिए जब फर्स्ट एप्लीकेशन हुआ था तो इंटरमीडिएट डेंसिटी थी, मतलब एक स्ट्रैंड पुराना था, एक स्ट्रैंड नया था. तो अब ये दोनों स्ट्रैंड वापस से सेपरेट करेंगे. सपोज ये आपका एक स्ट्रैंड है. लेट्स चेंज द कलर. दिस इज योर सेकेंड स्ट्रैंड. ओके. Now after this, these two strands they will separate. एक strand ये हो जाएगा, तो ये आपका दूसरा strand है. So both these strands will separate जब second replication process होगा, isn't it? Both will separate. Clear? Yes or no? Let's take the same colors. We are taking different colors. You might confuse yourself. It will become confusing for you. Let's take the same colors. I'm taking the red and blue only. So let's take the red and blue. Let's suppose, so this is your red, okay, and let's have the blue. So this one is there, okay. Now your second replication जब होगा तो दोनों strand separate करेंगे. So you have blue, okay, and next is you have red. तो आपकी red भी हो गया और आपके ब्लू, दोनों स्ट्रैंड सेपरेट हो गए. अब हमारे पास देखिए किस सोल्यूशन में E. coli मौजूद है? ओनली रेड सोल्यूशन में. ठीक है, ओनली इन रेड सोल्यूशन. तो जो भी नया DNA बनेगा वो रेड कलर का होगा. न्यू DNA विल ओनली बी ऑफ रेड कलर. ओके, न्यू DNA विल ऑलवेज बी ऑफ रेड कलर. सो दोनों स्ट्रैंड सेपरेट हो गए हैं. अब यहाँ पे एक कॉम्प्लीमेंट्री DNA बनाया जाएगा, वो रेड होगा. दिस ऑल्सो विल हैव रेड. ठीक है. सो दिस इज वन मॉलिक्यूल ऑफ DNA. अब देखिए हमारे पास 2 मॉलिक्यूल्स मौजूद हैं यहाँ पे, 2. सो आई हैव टेकन 1. ठीक है. सेकेंड विल ऑल्सो हैव द सेम. तो हमारे पास 2 रेड बनेंगे. 2 विल बी रेड, completely red, and 2 will be intermediate, isn't it? That is why you will have density at the 2 will have the lightest density and 2 will have the intermediate density. Clear? So 2 जो है हमारे रेड कलर के हैं completely, क्योंकि उनकी डेंसिटी वो लाइटेस्ट है, एंड 2 आर ऑफ मिक्स रेड एंड ब्लू कलर क्योंकि उनकी डेंसिटी इंटरमीडिएट है. क्लियर एवरीवन? इज इट क्लियर? ओके. Now let's revise it. So parent DNA was having N15 both, right? Then first generation जब पहला replication हुआ तो both strands will separate. तो 15 will have separated. Now new strand will be made in N14 and N14. ठीक है? This was in first phase, first replication phase. Now these two both will separate. They will also. So one will be 15, one will be 14, another 14 and 14, then 14 and 14, and 14 and 15. So these two strands, they are old. Okay, first. So now we have 2 completely new red. 2 will be completely red and 2 will have the mixture. They have an intermediate density. Clear everyone? Yes. So this will be your homework. First of all, you will complete the notes of this topic. And you will read the page numbers 100 to 105. ठीक है? So आप ये notes complete करेंगे, ये page numbers read करेंगे, and you will underline the important points. And if there is any doubt, you will work your work. You will send your doubts to me. Clear everyone? Okay. Bye everyone.